And well, first of all, I want to thank the organizer for giving me the opportunity to present my work here in this nice conference. And yes, the title of my talk is "Does Toxicity Promote Coexistence?" And I will go a little bit against the current today, as we have seen a lot of mathematics in this morning, but I'm not going to give a lot of mathematical details in this talk. Rather, I'm going to talk a little bit about the biology behind this work, at least in the first part of the talk. And I'm going to introduce the biological motivations that led me to study this problem and the phenomena, the biological phenomena that I'm trying to model and that I would like to try to explain through mathematical modeling. So the first part is going to be about biology, and then I will introduce the model I'm studying, and at first it's going to be just a model of ODE, and then I'm going to introduce diffusion in the system and just present this, some numerical results and some numerical simulations. So, okay, the phenomena I want to discuss is like harmful algal blooms, or HAB in short, which is phenomenon of great concern, especially nowadays. And this, the term HAB indicates is actually rather might be rather confusing terminology, because it indicates it's a general term to say a bloom of an algal bloom which it has negative effect on other organisms, and whether this is via the production of toxins or by mechanical damage or any other means. So HAB is rather like a generic term. And well, HABs includes different type of taxa, such as Dinoflagellates and Diatomaceae and cyanobacteria, which is the latter one is well known as blue-green algae. So, and this latter one in particular is a special concern because it has a potential impact on drinking or recreational waters, especially in lakes and rivers. And in fact, these cyanobacteria are able to produce compounds which are hepatotoxic, which means they affect the liver, or neurotoxic or dermatotoxic, so affecting the skin. And in this talk, I want to focus especially on this cyanobacteria and blooms caused by cyanobacteria. So, cyanobacteria they actually rank among the oldest living organisms, like up to 3.5 billion years ago they were present, and they were tremendously important to shape the atmosphere, and they were responsible for they are thought to be responsible for the atmosphere we're living in now, and for actually creating life on Earth as we know it nowadays. And well, regarding the algal blooms, like freshwater cyanobacteria blooms may be dominated by either single species or can be composed of a variety of toxic and non-toxic strains. And the great variety of these cyanobacteria can produce a toxin which is called cyanotoxin, and the most common species able to do so are like Microcystis, Anabena, and Planktotrix. But the Microcystis is the most abundant and most common bloom-forming genus. So it's the one I'm going to focus on, this particular cyanobacteria called Microcystis. And as I previously mentioned, like this Microcystis can be a major hazard in recreational lakes. And actually, I was looking around, like while doing research for information, and I ran across many websites, in particular also on the website of the EPA, of the Environmental Protection Agency of the US. If you go and look around, and you can find a lot of information about this guy here, because it is actually quite a serious problem. So, like, there is a lot of information about what to do and about avoiding drinking or bathing in lakes which present blooms. And in fact, these blooms can produce this hepatotoxin, which is called microcystin, which is poses serious health risks for both animals and humans. And in general, microcystis consists of a mixture of both toxin-producing and non-toxic producing strains. So there are strains which are able to produce toxin and strains that do not. And these two strains compete for resources such as light. So now I'm going to introduce an recent experiment performed by Edwin and other people and collaborators, which was a competition experiment for light between these two different strains of Microcystis, between a strain which was toxic and a strain which was not. And what they observed is that the non-toxic strain always outcompeted the toxic one, even when they gave the latter one a strong initial advantage. For example, here in these figures, you can see the result of the experiment, and as you can see, in like the open circles is the toxic strain, and the closed circle is the non-toxic one. So even when in the experiment they gave a big initial advantage to the toxic strain, this is proportion of 9:1 as in the initial state. No matter what, over time, like the toxic strain was always outcompeted by the non-toxic one. So this suggests that non-toxic strains are better competitors for resources. And well, now I'm gonna talk about the other guy I'm interested in this talk, which is zooplankton. Of course, all these cyanobacteria I've been talking about so far are what we also can commonly call part of the phytoplankton, which is the vegetable part of plankton, so plants, and which is formed by many different species and types, and cyanobacteria is just one of those types. While zooplankton are heterotrophic plankton that graze over phytoplankton, so they are predators for phytoplankton and for cyanobacteria. And in particular, this Microcystis may have a toxic or at least a non-nutritious effect that inhibits the growth of the zooplankton. There are also studies that show this. However, the zooplankton graze on these species despite their toxicity. So zooplankton keeps eating the toxic strains of Microcystis despite their toxicity. And so, however, like also some experiments, as one might intuitively think, some experiments suggest that in general zooplankton is able to select between these two prey and shows a preference, of course, for the non-toxic species. So, but still, the interesting thing is that they still also hunt and graze, let's say graze over the toxic species as well. So, and this is also another experiment that was done by a group of Chinese researchers like Wang and other, that in a lake in China, and they basically suggested that selective predation by zooplankton over these toxic Microcystis may actually promote the formation of toxic blooms. In fact, they performed these experiments like where they basically performed the experiment looking at the state of the water in a portion of the lake, and in the beginning they observed that so they performed several experiments, as you can see, like they added plus Z plus N, for example. It means they added zooplankton and added like nutrients to the lake, and in the beginning, or for example minus Z here, maybe it's not really clear, these stripes is where they removed zooplankton. So this is like the initial state, and here you can see the Microcystis is what we're interested in. So it's a really low percentage of Microcystis compared to the other phytoplankton species. So what happened when they actually at day 12 here, like after they added zooplankton to the system, You can see that the Microcystis increased a lot. Here, this shade of gray is where the zooplankton was added, and or where the zooplankton was removed. Here, like for example, here was removed zooplankton but added nutrients, and here was just this short column here is where they just removed the zooplankton from the system. So you can see there's a huge difference in the abundance of toxic Microcystis and that depended a lot on the presence or on the absence of zooplankton. So this suggests that the predatory activity of zooplankton may actually help to form blooms, because when the zooplankton was not present, they did not observe any blooming of toxic Microcystis. So now what I've been talking up to this point, like some questions do arise quite naturally. Like the first one is, does toxicity actually promote the coexistence of different species? Because we saw that the toxic strain was actually a poor competitor for resources. So without zooplankton or without any other thing, they were going to die. And also the other one, there's a mistake, I don't know, there's like 2-1, this should be a 2, of course. So that if does actually zooplankton's predation promote the Microcystis bloom formation? So it might be the predation help to trigger the formation of these blooms or not. So before introducing the mathematical model to study this phenomena, I make some assumptions which follow quite naturally after the experimental observation and results I introduced. So the first one is rather obvious, which is without the toxic prey, predator and the other prey, the non-toxic one, they coexist like normal, natural, like predator-prey situation. And this is like really common, like in plankton dynamics, like we know that zooplankton and phytoplankton coexist. So the second assumption is that without the non-toxic prey, predators go extinct. So if predators can only choose between a toxic strain, then they're slowly going to die, because the toxic one, as we saw, as I said, has an inhibitory effect on the zooplankton. And the last assumption is the one following from the first experiment I introduced, which is that without predators, so if we think about the two-component system consisting of two prey, the non-toxic strain is always going to win. So it's always a superior competitor for resources. So given this assumption, I introduce first a model of ODE. This is just a very typical and well-studied system of like Lotka-Volterra type with two competing prey and one predator. And this schematized simply by this simple diagram here. So we have two prey reproducing logistically and competing for resources, and a predator which feeds over these two prey. So this is the normal situation. While in our case, we have a slight but important difference, which is that this interaction with TPs for toxic prey and NTP for non-toxic prey, this interaction here is negative. So we have a negative sign here instead of the usual plus, because we, as I said, the toxic species has actually an inhibitory effect on the zooplankton. So we have basically this minus sign at first. So after some scaling and some rewriting the system, this is the final form of interest. And the two most so we have like here, this first part is like the competition between the two prey with A and B, and of course by assumption A3 we will choose A smaller than 1, smaller than B, so that X is a weak competitor for resources. And the interesting parameters are of course this D and mu, which is somehow the strength of the toxicity of the toxic strain, and D mu is the grazing pressure of the zooplankton on the toxic. Of course, intuitively, as we think, like as I said, there are experiments that suggest that zooplankton is able to detect the toxic prey, and So it makes sense to consider this d depending on mu. So, and the simplest choice I could think of was this one, to take this d mu as something like 1 over 1 plus mu. So what does it mean? That it means that if the prey is really, really toxic, so if this term, as mu grows, this becomes smaller, so the predator is going to prefer more and more the non-toxic one if the toxic species is really toxic. Otherwise, if mu goes to 0, then the predator makes no difference in hunting either the first one or the second one, because the toxicity is irrelevant. So now our biological questions, now the first one, it now translates in the following way. So does increasing the value of mu promote the coexistence of 3 species? And okay, so we have 3 species coexistence point, which is, of course, there are so many parameters, so it's rather complicated, really messy form, not really nice. And but it's rather easy to show that this equilibrium exists in the positive region of space if these 2 conditions hold. So if mu is bigger than this quantity, and if b is bigger than k times r. So if mu is actually smaller than this quantity, then this 3 species equilibrium does not longer exist, and it collapses on the equilibrium of without where the toxic prey is 0. So we only have non-toxic prey and predator. So a little bit, because of the assumption I made, it's rather easy to classify the equilibrium points of the system, and to say that, of course, like these first 3 equilibrium points, like trivial and semi-trivial points, are always saddles, are always unstable. And the reason is that, well, the first one is obvious, and the other 2 kind of also, because, for example, the third one, we said that in the 2-component system, this point 0 kr is unstable because the other one is always winner, so this is going to be unstable in the 3-component system. And this one is a saddle because we require that the predator and prey coexist, so we must have this relationship. R must be bigger than 1 over k. So this makes this point a saddle. What we cannot say is nothing about E3 and E4. First, because E3 in the 2-component system is stable by assumption. We assume that P and Z coexist. And E4, of course, is complicated. We cannot say nothing. And of course, by linear stability analysis, it's rather easy to show that E3 is always stable for any mu if this rather stringent condition holds. On the other hand, if this is not true, so if kr is bigger than b, then E3 is stable when mu is sufficiently small. So so when this condition holds, then this point might lose stability depending on mu, and as toxicity increases, it becomes more likely more likely for the toxic prey to invade into this system. So what happens when E3 is unstable? That's the question. And here I'm going to rely on some numerical analysis, because as you saw, like the form of the coexistence equilibrium point, 3 species coexistence, was say very complicated because there were so many parameters. So I'm going to fix some of the parameters and leave mu, which is the most interesting to our purpose, free. And I'm going to try to investigate the global structures of the equilibrium, like using auto, like using the software auto. And so, of course, these parameters are fixed so that our assumptions are satisfied, of course. So what happens? So the global structure is something like this. It's a rather simple, like, bifurcation diagram. So we see that when mu, this on the horizontal axis, we have mu, so this toxicity parameter here, we have the max of x in this case. So if mu is small, then the E3 equilibrium, so the equilibrium without where the x is 0, is stable. And as mu increases and it reaches a certain threshold, which is this b minus 1 over kr minus b, we have a branch point, and we have a new then this new stable branch, the equilibrium A4, they appear, so this 3 species coexistence equilibrium, and it's stable. So this means actually that increasing the toxicity actually leads to a stable three-species coexistence equilibrium. And maybe I want to make a short comment here, because as we've just seen, like if mu is small, then there is no chance for example, the toxic prey to invade into the system if we think about invasion. Or on the other hand, like a strong toxicity allows the three species to coexist. So this might seem a little bit paradoxical at first, because like we think of a species who's really toxic and the predator is still grazing on it. So we might think the predator actually gets a lot of damage from this prey, but nevertheless they coexist. But however, if you think it's important to remember that I choose the grazing parameter as something like this. So if mu increases, it's also true, we have to remember that the predator prefers more and more the non-toxic species. And also, if coexistence seems strange at first, we can explain like intuitively and roughly speaking in this way. So if we have x, if x is increasing, then we know that the predators z is going to decrease. But if predators decrease, then we have the population of the non-toxic prey is going to increase. And so at the same time, if they increase, they are better competitors for resource, so they outcompete x, so x is going to decrease. And so we go the other way around, and so z increases, which means p decrease, which makes x increase. So we have this sort of diagram. This sort of, this is really roughly speaking, of course, sort of cycle, but this gives an intuitive idea of why coexistence works or might work. And also think about the toxicity as a kind of the advantage that X, the Microcystis gets from the toxicity is twofold. Like one is direct, so they directly inhibit the growth of predators. The other one is a sort of indirect advantage, because they kill predators. So and so predators, as I said, they will prefer the non-toxic species, so they will hunt more non-toxic phytoplankton, and in some sense they will reduce the competition pressure that X suffers from P. So in some sense they have this sort of indirect advantage as well by killing Z. So now I'm gonna switch a little and also go back to actually Ohio, like the talk of Professor Luby we heard this morning. So because, as I said, like especially in the United States, but all over the world, like this HAB phenomena, especially these blooms formed by cyanobacteria, is rather a serious problem. So like this is a lake in France, this is actually Ohio River, and Iowa Lake, and also still in Ohio, like Grand Lake, St. Mary, and Lake Hope. There's a lot from Ohio, actually, and Gibbs Lakes in Wisconsin. So these are all like patches of where you see this green. This means like blooms of toxic cyanobacteria. So about we're interested also in the second question to answer if the predation by zooplankton might actually promote the formation of these toxic blooms. So I'm gonna allow the species to move around. So I will add diffusion to the system and consider the same problem with diffusion. And of course we consider like zero flux boundary conditions here. We're thinking, for example, about the lake, and also I have to spend a couple of words about diffusivity of plankton. So how plankton diffuse. First of all, like usually there are some works about plankton dynamics, but mostly, as far as I know, they're most about marine plankton from the sea, from marine plankton, and they usually, some of these works like consider negligible the movement of plankton compared to the turbulences of the sea, of the currents. So, but here we're interested in freshwater planktons in lakes. So I think it makes sense to consider different diffusion coefficients if we're in a situation where we don't have where the water is calm and we don't have particular currents. And in particular, like zooplankton, it's quite bigger, first of all, than phytoplankton, and swims much, much faster. I mean, while phytoplankton and cyanobacteria are much more like gliders, they simply move slowly on the surface. Zooplankton moves pretty fast. They actually of the order of millimeters per second, and they're pretty quick. So I will consider like D1 and D2 to be the same because we have the same Microcystis, and phytoplankton, and D3 is going to be considerably bigger than these two. So what happens? I'm going to have some simulations. Okay, so here I fix the diffusion parameters in that way. So on the left we have, so the red one is the toxic Microcystis, and the green one is the non-toxic, and the blue is predators. And they started steady states here, but in this case mu is slightly bigger than the critical mu we saw. So in the ODE system, like this point is stable. And so what happens if we fix mu in this way, just slightly bigger than mu c? We see that even if despite I gave quite a disturbance in the middle, so I increased quite a lot the density of toxic Microcystis here, they just smooth down to equilibrium again. So in this case, like even in the PDE, like this equilibrium seems to be stable. But what happens if we increase mu a little more? So if we increase mu slightly further, so I go into 0.2, and it takes some time to kick in, but we can see we have a sort of Turing instability, due to diffusion and the formation of this in one-dimensional case of this pattern. So we observe we have regions where the toxic cyanobacteria Microcystis actually grows a lot, and the non-toxic one decreases, and also like we have this oscillation on predator. So but the important thing is that in these points we have these spikes of toxic cyanobacteria. Well, here I'll show you a two-dimensional simulation, which is, how much time do I have? Just to...
We've got about 2 minutes.
Okay, perfect. So here we have this. Okay, so also started at equilibrium, and the initial condition was like randomly giving some, increasing a little bit, just a little bit, the density of toxic Microcystis again. So always here like it's a little slow, but well, always also in this case we can see that again, like the colors are the same. So actually now green, red is the toxic species. So we can see that even some time we have again these points, these patches where we have a high concentration of toxic Microcystis. Okay, so now maybe I should have used green for the toxic one, but as we can see, like we have the emergence of these patches in space. So the simulation shows that, as I said, we have also the formation of these patches which are observed in nature, like for example in these cases. So like my time is up, but anyway, I've finished. I want to give some conclusions. And as I said, like this Chano HABs is a widespread phenomenon, and that causes a lot of concern because it's actually seriously could be potentially toxic not only to animals but also to humans. So I think understanding the dynamics that govern such phenomena is of great importance. And well, among the studies there are a few mathematical studies on HABs, but as I said, they're mostly focusing on plankton on the sea. No one is doing like cyanobacteria, and they always use the underlying assumption, because they don't say it clearly, that the two species of toxic and non-toxic phytoplankton, they coexist without predators, which I don't know. I question this because, well, there is, at least for cyanobacteria, there is more than an evidence and more than some paper that suggest that the non-toxic species is always a better competitor. So here I built this model in order to try to understand how this bloom work. And well, the results so far are promising, because they're quite in accordance with the experimental results, as the one from the Chinese group, that they were suggesting that the zooplankton activity might actually trigger and or promote the formation of this toxic bloom, because otherwise, without zooplankton, they did not observe any blooming. And also with this simulation, we've shown that we have these spotted-like patterns, which also we observe in nature. And of course, however, this research is ongoing, and it's what I'm doing now for my PhD. So there is still a lot of work to do, especially also to try to prove rigorously some mathematical results. So there's a lot to do and can be improved. So this, well, thank you for your attention. I hope it's correct.